Hi everyone, welcome to my channel Microbiology with Sumi. If you like my video, please like, share and subscribe to my channel. Today's topic is cyanophage and its details. Before starting with cyanovirus, let's see some details about cyanobacteria. Cyanobacteria are microscopic, photosynthetic and aquatic bacteria. That is, they can be observed under microscope. They are photosynthetic, that is, they synthesize their own food, and they are aquatic in nature, so aquatic bacteria. It is generally found in fresh water, lake, ocean, streams, moist soil and rock surfaces. Cyanobacteria is also called as blue-green algae and also known as cyanophyte. Now they are aquatic, photosynthetic, prokaryotic bacteria, so its name was renamed from cyanophyte to cyanobacteria. Cyanobacteria have internal membrane and flattened sacs called as thylakoids. Now this cyanobacteria have internal membrane like normal bacteria, but they contains flattened sac and these sacs are called as thylakoids. Now in thylakoids, photosynthesis process is carried out. Now this is a place in the cell where photosynthesis is carried out. Cyanobacteria are able to fix atmospheric nitrogen and hence can be useful in agriculture as biofertilizer. So they have an ability to fix atmospheric nitrogen. Now examples of cyanobacteria are spirulina, nostoc and anabina. There are many more examples, but here I have given you some examples. Cyanobacteria are microscopic in nature, but its colony may be large enough. When its colony grows, it causes large blooms. Now, when we observe a cyanobacteria, we require a microscope for its observation. But when its colony grows, this colony is large enough. It causes a layer on the surface of water, and this layer is called as blooms. The blooms may be harmful to human beings, plants, animals, and aquatic life. These cyanobacteria forms a layer on the surface of water in various colors like green, blue, brown or red. So it is called as blue-green algae. This layer causes the depletion of oxygen in the water and affects the aquatic life. So if the oxygen will not dissolve in water, how will the aquatic plants grow? And if the aquatic plants will not grow, how will the fishes get their food? So it is important that oxygen should get dissolved in the water. It may also release some toxins in the water which may affect the taste, odor and aquatic life. Further, let's see cyanophage. Cyanophage is a virus that infects cyanobacteria. Here phage means the bacteria that infects the bacterial cell. So the virus which infects the bacterial cell is called as a phage, and cyanophage is the virus which infects the cyanobacteria. Cyanophage uses cyanobacteria for its replication and multiplication. Cyanophage is classified in a bacteriophage family, that is Myoviridae, Podoviridae and Siphoviridae. Cyanophage was first discovered in 1963 by Safferman and Morris. They discovered the first cyanophage and named it as LPP1. So there were two scientists, that is Safferman and Morris. In 1963, they discovered the first cyanophage, and this cyanophage, that is this virus, was named as LPP1. The three cyanobacteria, that is Linkbay, Plectonema and Formidium, were attacked by this virus. So this virus was named as LPP1 by using the first three letters of the genera. So this LPP1 virus attacked these three cyanobacteria, and initials of these three bacteria are used for naming this virus. Further, many serological strains of LPP virus were isolated from all over the world and hence named as LPP2, LPP3, LPP4 and LPP5. Now the eutrophic lake and polluted water supports the luxurious growth of cyanobacteria. Now this eutrophic lake and polluted water provides a large number of nutrients for this cyanobacteria and hence support its luxurious growth. Now hence cyanobacteria form a bloom which can result in mortality of aquatic life. Now luxurious growth and luxurious metabolism results in formation of a bloom of cyanobacteria on the surface of water, and which results in mortality of aquatic life. So here cyanophage helps in controlling such blooms. Now here cyanophage plays an important role in controlling such blooms of cyanobacteria. Let's see morphology of cyanophages, especially LPP1 group viruses. Cyanophage has icosahedral head and short tail similar to T3 and T7 viruses. They may differ in morphology as well as physiochemical property. So there are several cyanophages and they may differ in their morphological characters as well as physiochemical properties. Now, tail of cyanophages may be contractile or non-contractile. And one group of cyanophages are similar to T2 and T4 phages. Group G3 and D1 of cyanophage are serological similar to T phages, but there is no relation between them. Now, let's see growth cycle of cyanophage. Cyanophages show one-step growth curve. The growth curve of cyanophage is similar to T4, but the latent and rise period is different. So the growth curve is completely similar to T4, but the latent period and the rise period is different as compared to T4. Multiplication of LPP1 is carried out in the following manner. Adsorption. Virus is attached on or absorbed on the surface of host cell. So this is the first step. The virus gets attached on the receptor sites and further the injection process is carried out. Now the second is injection. Genome of phage is injected in the host cell and protein coat is left outside the cell. Now DNA injection mechanism is unknown. Now in injection mechanism what happens, the genome of the phage is inserted in the host cell and the protein coat, that is capsid, it remains outside the cell. So here's a special, means in detail the DNA injection mechanism is unknown here. Reduced protein synthesis. As soon as genome enters, the rate of protein synthesis of host cell is reduced and blocked by the phage genome. Fourth is multiplication. The cyanophage starts to multiply. Fifth is assembly. After multiplication, the viral nucleic acid and protein coat starts to assemble to form a procapsid and then it is released. After assembly, the cyanophage get released from the host cell by lysis of cell. Let's see some important notes. Here after insertion of genome, three types of proteins are formed and that are earliest proteins. These proteins are formed immediately after genome enters. So as soon as the genome enters, the earliest proteins are formed. Second are early proteins. Now these early proteins are formed after 2 hours of entry of genome till lysis of cell. Further, late proteins. They are formed after 4 hours of infection until the host lysis. Further, degradation of host cell genome takes place after 3 hours of infection. And now this degraded DNA material of host cell is used for synthesis of viral genome. So, these early proteins I told you, earliest proteins, they form immediately. Early protein forms after 2 hours of infection. Late protein forms after 4 hours of infection. Further, the degradation of host genome takes place after 3 hours of infection. After 3 hours, now this degraded DNA material of host cell is used for synthesis of viral genome. The latent period of LPP1 and N1 is 7 hours. That is the complete period, we can say, from the infection till release is 7 hours in case of LPP1 and N1. The plaque forming units, that is PFU, may also vary from virus to virus. Further, burst size. It is a number of phages released after multiplication and replication of viruses from the host cell. So the number of phages which get released after multiplication and replication of virus from the host cell is called as a burst size. The burst size of viruses may also vary. For example, LPP1 is 350 PFU, AS1 is 50 PFU, N1 is 100 PFU, SM is 100 PFU. So this was all about cyanobacteria and cyanophage. So if you like my video, please like, share and subscribe to my channel, and thank you for watching. Please don't forget to comment in the comment section.